I'm really excited to talk to you about the cell raft technology and how it can really transform your cellular workflows, everything from 2D single-cell cloning all the way to organoids. So to start this discussion, I'm going to show you a montage of pictures from our own lab that I like to call cell-inflicted pain at the microscope. So I'm imagining that many of you here are scientists at the bench, or you're managing scientific programs. So we all know what it's like to walk into the lab, put your cell culture plate on the microscope, and then kind of have that gut-wrenching feeling. Why? Because of dead cells, empty wells, polyclonal populations, and if you're ambitious enough to take on organoid assays, well-to-well variability that is sure to impact your downstream assessment. So while we've all experienced these failures, we realize that they have a significant impact on our research, including our timelines, wasted resources, as well as oftentimes our overall project success. So today, I'm going to introduce you to a new way to culture your cells to improve viability, how to get more of your desired phenotype with less effort, and how to unlock the potential for reproducible organoid assays. This all starts with a new way of culturing your cells. The cell raft array is an innovative culture plate. Cells are seeded onto the cell raft array, much like you would seed any other tissue culture consumable. Importantly, these cells also share a contiguous media volume like they do in any flask or dish that you're culturing your cells in. But importantly, within the reservoir of the array, we fabricate an elastomeric microwell array with thousands to tens of thousands of these microwells. This provides spatial segregation of thousands to tens of thousands of cells within the reservoir. At the floor of each microwell is a microscale polystyrene cell raft. These cell rafts are releasable from the array. And just to show you with a little bit more detail what this looks like, so here in white, you can see the microwell array. At the floor of the microwell is this magnetic polystyrene cell raft. And your cells attach and adhere to the cell raft just like they do any other polystyrene tissue culture consumable. But as I mentioned, really the key feature to the cell raft array is that while these cells maintain spatial segregation and can be seeded on a single cell level, they're sharing that contiguous media volume, allowing them to exchange growth factors, which ultimately has a significant impact on your cell viability and the growth of your cells. So as I mentioned, the cell rafts are releasable. So this animated video is going to show you what the process looks like. So here is my desired cell of interest adhered to the cell raft. A needle penetrates the floor of the cell raft array, releasing that cell raft from the microwell. Because they're doped with magnetic particles, we pick up that cell raft and transfer it to a 96 well plate with a magnetic wand. Once the cell raft is in the 96 well, cells passively grow off that polystyrene cell raft onto the 96 well plate. So here you can see we have the video of the colony outgrowth over time. So today I'm going to discuss the ability to leverage this technology using 3 case studies that I like to call the ineffective flow sorter, the inefficient iPSC, and the inconsistent organoid. So when we're culturing our cells using standard methodologies, our cells are in their natural environment, they're in bulk culture, they're exchanging growth factors. But if we want to clone, edit, or manipulate our cells, the first step is to get them into single cell suspension. Beyond that, we're either using technologies or limiting dilution to put a single cell in every well of a well plate. So this process of going from your bulk culture to your single cell culture removes cells from their natural environment and ultimately significantly impacts your viability. This process is further compounded if you're using instrumentation with fluidics, ultimately causing a reduction in your process, both with the inefficiency. This process can be very manual if you're using limiting dilution, or incredibly expensive because you're using some other technologies, or lots and lots of consumables, media, and reagents to get as many clones as you want. And then at the end, a lot of times what you have are fewer single-cell derived clones, and the process is just very time-consuming. So in our first case study today, I'm going to present to you a partnership with an academic collaborator from Duke University. His lab was interested in generating a stable expressing cell line with two independent constructs. The first construct was a blue fluorescent protein. The second was human epidermal growth factor 2, or HER2. So the important part of this is that their process of finding their double positive cells involved both antibody staining as well as flow sorting to find the double positive cells. So their approach with the technology they had available was to do antibody staining and flow sorting. On their first sort, they had, I think you can see this, roughly 10% of their population was double positive. They took that sorted population, put it into culture, expanded it out, and did a second sort of that initial sorted population. On the second sort, 89% of their population was double positive. So this process took multiple rounds of sorting. It was very time-consuming for them. And ultimately, at the end, what they were left with is after multiple passages, despite that 89% positivity on their sort, less than 10% of the population was actually double positive. And that's likely because those BFP positive cells that were not HER2 positive outcompeted the double positive cells as they were performing their passaging and banking and pulling those cells back from cryopreservation. So we were able to take his sorted population, transfer the workflow to the cell raft array. So here you can see in the top panel, we can very clearly identify polyclonal cell rafts. So here you're looking at two cells. Both of those cells are BFP positive, compared to our monoclonal cell rafts, where we have a single BFP positive cell. We let these cells grow on the cell raft array until they become colonies, and then perform antibody staining for that HER2 expression. So here you can see in the top panel, we have very few cells from this polyclonal cell raft that are actually HER2 positive, even though both of those cells were BFP positive. Whereas when we're able to select this monoclonal raft, all of the cells from this population are in fact HER2 positive. So we took this a step further because we wanted to quantitate the efficiency of the cell raft array for this process. So in this table, you're looking at the number of clonal cell rafts, so single cell derived clones on the cell raft array. 444 rafts were found to be BFP positive but HER2 negative, and only 76 rafts were found to be double positive. So we isolated some of these double positive clones. And here on the top, you can see the timeline of our workflow. They were on the cell raft array forming colonies for 5 days. We performed that antibody staining. We isolated those clones and then grew them out and cryopreserved them. Lastly, to assess the efficiency of our process, we took those cryopreserved clones, put them back into culture, and did standard antibody staining for the HER2 expression. So here you can see we have a nice double positive clone after the whole banking and reinitiating into culture process. So overall, using the cell raft array, we generated 27 stable double positive clonal cell lines for our collaborator. This represented less than 1.5% of the total population we were able to screen on the cell raft array, and we did this in 3 weeks. So just like these clonal cell line development processes plagued our academic collaborator, they're also a big problem in big pharma. So here you're looking at one of our customers' workflows in their cell line development core. So here you can see that the whole process for them is roughly a 13-week timeline. And what I'd like to point out here is that the part of their workflow that takes the longest and that they have the longest buffer of time is their clone generation. And that if that's not telling that their process is slightly inefficient or unpredictable, we're able to do this, as I showed you previously, in a much shorter time. So this customer was working with suspension cells in particular. And because they're a cell line development core, they actually perform direct comparison to other instrumentation to look at the efficiency of their cell line development workflow. So overall, what they found is that secondary sorting of their suspension cells was very harsh and inefficient. With the cell raft technology, they were able to improve the viability of their cells. They really enjoyed the feature that they could monitor their clones over time and confirm that they were clonal. They found the cell raft array to be incredibly easy to use, and were actually thinking about a direct edit to array workflow because of their success. So here you can see some of the time course images that they shared with us. We're looking, again, we were coding the arrays to do suspension cell cloning, and in one experiment, they were able to generate over 400 clones. Some of the benefits of the cell raft array that they mentioned specifically were that the workflow led to shortened timelines and greater efficiency. So our next case study is the inefficient iPSC. So for those of you who have worked with iPSCs, you know that even traditional methods of culturing iPSCs can be very challenging. It takes really well-trained hands, and oftentimes, even with standard culture, you're experiencing poor viability. If you are doing clonal workflows, it's really challenging to find clonal iPSCs in standard culture. The media and additives for media are incredibly expensive, and because of the sensitivity of these cells and frequent feedings, you're often running through a lot of reagents through your process. Also, in the process of manipulating these cells, you oftentimes have a loss of pluripotency. And if you are doing some sort of clonal, manual cloning, this is a very laborious process in the lab. So, as I mentioned, growing iPSCs from a single cell is really challenging. With the cell raft array, as I showed you in the previous case study, we're able to really track and trace that single cell to clone. So here in the panel, you can see we have 4 different iPSC lines, and the arrows are pointing out single cells at time 1, and we can watch those cells form nice colonies on the array throughout the duration of our experiment. Importantly, we're also able to screen over 500 times more cells per consumable. So here down in the bottom, this is quantitated in a panel where we're comparing how many cells we screen on the cell raft array versus in a single 96 well plate if you were doing limiting dilution or some other technology where you're putting a single cell in a well. We've also shown that the cell raft array is compatible with any iPSC coding, and you're also using over 1,000 times less coding per cell screened because of the volume in the array and the micro-well features compared to just coding 96 well plates. Similarly, we've also shown compatibility with several different iPSC medias, and overall, again, you're using 2,000 times less media per cell screen compared to 96 well plates. So this process is really very efficient. You're able to generate thousands of clones on a single consumable using less reagents throughout the process. So importantly, we can also streamline one of the most important steps of iPSC cell line development, which is conformation of pluripotency. So here you're looking at colonies of iPSCs grown on the 200 micron cell raft array. You can see that we've done on-array antibody staining for TRA160, so we can confirm that the cells that we're isolating are indeed pluripotent. As I mentioned, the workflow is really very efficient because compared to other technologies, we aren't isolating a single cell in a well like you would with a sorter or droplet dispenser, or even if you're doing manual limiting dilution. So using the imaging features, we can track this single cell to iPSC colony while it's on the array, allow that single cell to grow into a nice confluent colony before we isolate into the 96 well plate. And then you can see here in just 2 days we have a nice colony growing off of that cell raft with the canonical iPSC morphology with tight borders and really nice cytoplasm to nuclear ratios. So to sort of prove the point, so to speak, that the cell raft array is much more efficient because of this contiguous media and culture environment, we wanted to do sort of a comparison of iPSC clone generation to limiting dilution, but also isolate cells at different stages on their growth curve from the cell raft array. So you can see here that when we isolate single cells on a raft, we get a very similar outgrowth efficiency as limiting dilution, right? Because cells don't like to be alone in a well. However, if we wait until roughly the forest cell stage, or even better, the small colony stage, our outgrowth efficiency just goes through the roof, right? So you can imagine the impact that this would have if all of a sudden you're able to get more than 90% outgrowth from your iPSC clones that you're interested in. So I've just shown you some on-array expansion and characterization. The next couple of iPSC workflows that I want to show you are on-array reprogramming and differentiating iPSCs into organoids. So more recently, we've been able to demonstrate opportunities to improve manually intensive reprogramming workflows using the cell raft array. So up here on the top left, the contrast might be low, but the arrow is pointing you to a single electroporated fibroblast on day 1 of the reprogramming process. You can see here that through serial imaging of the array, we're able to watch that fibroblast as it continues to grow. And then roughly right around here, maybe day 11, you can see that we start to get a morphology shift in those cells that looks much more like a compact epithelial or reprogrammed iPSC. And then by day 15, we have this nice confluent colony. We do TRA160 staining and see that it stains positive, which is good evidence that this is a clonally reprogrammed iPSC colony. So we can take that raft for isolation and now expand a pluripotent colony in our 96 well plate. So we've actually done this side by side again in the traditional methods using a 6 well plate. So with the traditional workflow, when you're ready to move these reprogrammed clones around manually, ultimately you get something that looks like this, right? So I have a nice iPSC colony here, but I have a lot of contamination, so to speak, of my non-reprogrammed cells. So this is that cell raft that I was just showing you in the previous images that we stained for TRA160. And you can see in just 11 days off of the array, we have this large, beautiful iPSC colony that has grown off of the cell raft. So again, we're scientists. We like to quantitate things. So to highlight the efficiency of this workflow, what we did is we counted in 3 wells of a 6-well plate versus 2 cell raft arrays. We counted the number of TRA160 positive foci, and you're looking at an average here in the table. So the average for a single well in a 6-well plate was roughly 440 positive events. Whereas on the cell raft array, we had over 1,200 positive events, which is nearly 3 times the number of reprogrammed colonies on the array. And that, tied with the ability to perform automated isolation of those cell rafts into your collection plate, is really going to streamline this workflow. As the sort of closing to our iPSC discussion, I'm going to talk to you a little bit about some of the work that we've done with iPSC differentiation into organoids. So we've done this with several different commercially available kits for generating kidney organoids as well as neuronal organoids. So this particular one is a cerebral organoid kit. The workflow is really very straightforward, but what I'd like to point out here is that anytime we're doing organoid applications, we expect heterogeneity within our population. So to sort of highlight that, we intentionally co-cultured 2 different iPSC lines, one that had a green fluorescent reporter, one that had a red fluorescent reporter. And so you can see here on this field of view on the array, we have green organoids, we have red organoids, and then we have some that are dual fluorescent. But the beauty of the air system is the ability to sort of evaluate this heterogeneous population and look at those cell rafts on an individual raft basis. So here on the top, you can barely see the single red iPSC at day 1, but we're able to form a clonal organoid, or the start of a clonal organoid across these 7 days. That's red fluorescent. This is a green fluorescent that starts from a small fragment of cells. And then if you can see here, in this bottom panel, we have a single red cell and a single green cell that forms this dual fluorescent organoid. So with these images, I'm sure you can imagine the opportunity to be able to tailor your downstream organoid assays if you can interrogate or sort out these individual phenotypes from a heterogeneous population. So just to give you a little bit more information on these cerebral organoid workflows, similarly to what you were seeing here before, I have a single cell derived green fluorescent iPSC on day 1. On the array, I can do media changes for embryoid body formation, neural induction, and expansion. At day 10, I can find those organoids that I'm interested in, isolate them into maturation media in the 96-well plate, and then you can see here from day 10 to day 37, not only do we get continued organoid growth, but we get this really beautiful neural network growing off of our main organoid body. So this time course image stops at day 37. We've actually taken these organoids out beyond day 60 and see no reduction in viability over time. So you can imagine that with the ability to sort of characterize the population, continue to grow your organoids off the array, this really opens up a lot of opportunity for organoid assays. So to summarize the inefficient iPSC, I've shown you that you can easily track and trace single cells into colonies using the cell raft array. The culture conditions of the cell raft array improved iPSC clone generation more than 25% compared to limiting dilution. We're able to confirm pluripotency on the array, so you're only isolating those pluripotent iPSC colonies that you want. I've shown you we can significantly reduce your timeline, consumables, and materials with the process of clonal generation as well as reprogramming. And I've also shown you just a teaser of some of our 3D iPSC organoid work on the array. So our final case study this morning is the inconsistent organoid. So for those of you who have done organoid culture, you're probably very used to seeing an image or something through the eyepieces that looks like this. The challenge with organoid assays is that our standard culture methods in a matrigel dome require multifocal imaging requirements, so expensive equipment, as well as a lot of image analysis. The population, again, as we mentioned, is heterogeneous, both in organoid size, as well as things like viability inconsistencies. And then ultimately, when you're doing any sort of downstream assay, you're limited to pooled readouts. This can sort of mask any sensitive assay that you're looking at performing when you're looking at the whole population rather than individual organoid analysis. So using the cell raft array, we're culturing organoids with a completely different approach that I like to call outside the dome. So here you can see a stitched together image. This is every field of view on the 500 micron cell raft array. All of the green circles highlighted here are cell rafts containing an individual organoid. So you can see we can grow and maintain hundreds of individual organoids. Importantly, all of these organoids are on a single focal plane. We can serially image the organoids over time to get those nice time course images of organoid growth. And then you're really able to phenotypically characterize the whole population and identify individual organoids of interest that you would like to use for any downstream application. So to sort of present to you a drug screening type application, I come from a toxicology space. So I used mouse hepatic organoids. We used the cell raft array to grow and segregate our organoids. We isolated two different populations in parallel from the same array. One population was not selected on size, so these organoids were anywhere from 50 microns and larger. The other population we selected for size of roughly 300 to 500 microns. We treated those with a 6-point dose curve of acetaminophen, which is a canonical hepatotoxicant, with 5 replicate wells each for 72 hours. So our endpoints for this were kinetic viability, so doing a reading every 24 hours across the treatment, as well as using 3D cell titer glow to measure ATP after the 72 hours of exposure. So here you're looking at 72 hours post-treatment, that unselected population of organoids. So you can see here, again, just to orient you, these cell rafts are 500 by 500 microns. So this organoid here is probably roughly 2 millimeters in diameter, whereas this one is maybe 300 microns in diameter. So for those of you who have used these type of assays that are really reliant on cell number, you can imagine the havoc that this wreaks on your reproducibility, right? So using the cell raft array, we were able to select those organoids based on size, get much more consistency in our downstream assay, and just as you would expect, this has an impact on our assay as a whole. So here in these graphs, you're looking at 24, 48, and 72-hour cell tox. And then in red, you have the 3D cell titer glow. So of that unselected population, you can see we have these massive error bars, whereas with the size-selected organoids, our error bars are significantly reduced. We're actually able to calculate an ED50 from this curve, altogether showing you that just by size selecting these organoids, we're able to decrease well-to-well variability within our assay. And also, if you were doing multi-plate assays or wanted to do biological replicates where you were performing this assay multiple times over, you would be able to maintain this consistency across multiple different experiments. So just to wrap up the inconsistent organoid story, I've shown you today that with the cell raft array, we can grow spatially segregated organoids in a single focal plane. We can reliably image those organoids over time and select organoids based on phenotypic or morphologic characteristics for generating custom 96-well plate assays downstream. We are also ultimately able to reduce our well-to-well variability compared to traditional dome culture methods of organoids where you have a heterogeneous population. So now that I've shown you all this great data on how the cell raft array solves a lot of these cell biology challenges, I'd like to introduce you to the technology in a little more detail. So this is the broad overview of the cell raft technology. I've already shown you the cell raft array and how it provides flask-like culture conditions and single cell separation for your cells. The cell raft air system performs the automated imaging of cells grown on the cell raft array. We have image-based software-guided selection tools to help you find your cells of interest for phenotypic and morphologic characteristics. And then once you've identified the cell rafts that contain your cells of interest, the cell raft air system performs the automated retrieval of your cells, colonies, or organoids. So just to get in a little bit more detail of each of the parts and pieces, the cell raft air system houses an inverted microscope capable of bright field and 3-channel fluorescent imaging. We use a 10x objective to capture images of every single cell raft on the array. And using those images captured from the air system, we can perform in-depth software analysis with those images to help you find your cells of interest. So what does it look like when you have this heterogeneous population of cells? So here I'm showing you a diagram that should represent multiple channels of fluorescence here, so red, green, and then dual fluorescent being yellow. There's really 3 modalities that you can interact with that imaging data. The first is cell raft review. So this allows you to step through every field of view of the array, look at each of the individual cell rafts and your cells grown on the cell rafts over time. But really the brains of the operation is cell raft cytometry. So this is where you can specify parameters such as size, shape, or fluorescence to help you find the cells that you're looking for. And then maybe the unsung hero that I really like a lot is our cell raft organizer. That allows you to incorporate the cell raft cytometry populations as well as any sort of manual scoring you've done in the fields of view to overlay or build filter sets for multiple different criteria. So cell raft cytometry. Our cell raft cytometry uses advanced bright field algorithms, so you don't have to have fluorescence to find your cells of interest. Importantly, it is offline of the instrument, so you can be sitting at your desk while your colleague is scanning arrays or isolating arrays on the instrument, which is really important for our multi-user labs, and compared to some technologies that we're used to, doesn't tie down the instrument for a single person for a long period of time. So using our cell raft cytometry, you can define populations of interest. So here, maybe hard to see, I've highlighted a population for finding single cells. There is a list of parameters that are all adjustable to help you find your cells of interest for things such as area, size, morphology, and then, of course, you can add in those fluorescent filters. The population set feature allows you to overlay multiple populations that you've made. So, for example, if you're doing a clonal cell line development workflow, you might be looking for single cells at scan 1, cells that have doubled at scan 2, and then at your last time point or somewhere further in the experiment, a colony of cells at that later time point. But importantly, you can overlay these populations Select the intersect of all three of these, and now you have a generated list of all of the cell rafts that were a single cell at time 1, doubled by time 2, and had a small colony at time 4. So after you've identified your cell rafts of interest, you can very rapidly map them for automated isolation using the air system. So the air system, as I mentioned, performs the automated release and transfer of those cell rafts to the 96 well plate. So in the beginning, I showed you an animation of what this looks like, but I always like to show the real-time view. So here, again, to orient you, this is a 500 by 500 micron raft with an organoid on it. As I play the video, you'll see the focal plane go out of focus. That's the needle releasing that cell raft from the microwell. The magnetic wand dips in, picks up that cell raft while the organoid remains attached. After the wand has retrieved the cell raft, it transfers it to the 96 well plate at your desired location. So with the cell raft technology, we have shown very broad cell line compatibility. In our internal applications team, we have characterized over 100 cell lines, including primary cells such as iPSCs and organoids. We have run the gamut of your traditional production cell lines, as well as developed protocols to coat the array for suspension cell line development. As I've shown you today, the application areas for our technologies and that our customers are really finding the technology to be superior are really diverse as well. So we've talked a lot today about cell line development. We've also generated some direct-to-array CRISPR editing data. And then, of course, much of what I showed you today was stem cells. And if you were at the talk yesterday or have seen some of our other content, we have a lot more content generating organoids on the array as well. So with this, I'll just quickly summarize. I've shown you today that using the cell raft array, we can maintain healthy cells in a contiguous media. We can automatically screen thousands of cells on a single consumable and track and trace the single cell growth to a clone or organoid. Using the air system, you can easily isolate those cell rafts containing your single cells or clones, and ultimately, you're able to simplify a lot of these challenging, labor-intensive workflows using the technology. So with that, I will thank you for your attention, and I'll invite my colleagues, Emily Shepke, who is one of our field application scientists, and Scott McMenemy, who's our North American sales leader, up for our Q&A panel discussion. Also, if you'd like to see the technology in real time, we have an instrument at booth 2430, and we'd be happy to give you a walkthrough of the technology there.
Thank you, everybody, for your time today. Are there any questions on the technology? Yes, sir.
Just want to make sure I understand the practical aspects. Could you go to actually slide 25 with the iPSC workflow?
The iPSC organoid workflow?
Yeah. So up to day 10, are you in the Petri dish? And then at maturation, you move to the 96 well plate?
So, yeah, so from day 1 to day 10, we're on the cell raft array.
The array.
Right, so we have thousands of microwells on that array that have organoids that are forming just like this. So this is just one example of that time course image. At day 10, while those organoids are nice and round and they're the size that we want, we use the air system to isolate them into the 96 well plate, where we have a single organoid per well, and we can continue the maturation of that organoid off of the array.
When you're in a single organoid state, is there additional ECM there? The organoid can continue to grow, or does it always stay within
The raft, yeah, that's a great question. Yeah, so there's two parts to that question. So the first part is for most of our organoids, we do spike in an extracellular matrix suspension into the 96 well plate. For some organoid types, like your cystic organoids that have a hollow lumen, they're a little bit more reliant sometimes on needing that ECM. With the cerebral organoids you're seeing here, we spike in ECM while the organoids are small like this, but we're not adding ECM at every media change, right? So we're spiking in a low concentration of ECM at day 10 just after isolation, and then these kind of grow by themselves out to day 30 and beyond. And then the second part of your question was what happens to the raft? Yeah. That's also organoid dependent. The short answer is nothing. The long answer is I have seen that some organoid types really do have a cell to polystyrene bond with the cell raft. Other organoid types are sort of just using that ECM as a loose attachment to the raft. So what that means is that downstream in the 96 well plate, sometimes the raft stays with the organoid, like you can see here. So here in the, let's see if I can get my pointer out. Well, it's not going to let me do it. So here at day 37, you can actually see the faint cell raft here at the bottom. The orientation is now, vertical. Other organoid types, by this point, and we've done multiple media changes, the raft is somewhere else in the well, and the organoid is doing its own thing.
That's very cool. This is lovely.
Thank you.
The lady in the green shirt. Yep.
No. So, yeah.
So the question was that throughout the period of the 10 days, are the array kept within the cell raft air instrument, i.e., is it environmentally controlled?
So no, the array is only on the instrument for imaging and isolation. Otherwise, that array is going back to your incubator. The benefits of that are you can run as many experiments as you want to at one time. Imaging is really very rapid, so bright field takes anywhere from 5 to 8 minutes, depending on the array format. And then when you add in your fluorescent exposures, your time increases a little bit due to the exposure times, but typically we're imaging an array in under 10 minutes. Do we have a micro injector incorporated into the system? No. So there's no fluidics involved whatsoever. We are relying on gravity and a Poisson distribution to allow our cells to settle into the microwells.
Yeah. Hi. For suspension cells, I may have missed this. Do you have to coat the raft, or how does that work?
Yeah, so for most suspension cells, you can coat with poly-D-lysine, poly-L-lysine, polyolysine. We'll do a 24-hour coat or overnight, and that'll keep them stuck down until you go to isolate. And then once you isolate, you can either triturate them back into suspension, or as they divide, they'll naturally pop off of the raft and go back into suspension.
My question was also about suspension cells. I'm thinking about, as you have cell propagation still in the raft state before you release them, now they're coming off the surface again. I don't quite understand that.
I think that this comes down to strategy of timing as to how we work that. So the question is, with suspension cells, as they continue to divide, if they're not then contacting the surface, what is our strategy around ensuring that we, take that into account?
Yeah, definitely. So for that, with suspension cells and adherent cells, we recommend imaging every 24 hours or every doubling stage. And so you'll start to see if they do start coming off, you can always isolate earlier if needed. But that whole process of growing them on the raft is typically only 3 to 5 days before you get a sizable colony that you can isolate. And so by that stage, they're still stuck down to the raft very well.
Yes.
Yeah, sorry.
So the question is, what is the range of array formats and applications that we have?
Sure. So Allysa has been showing the ones for organoids, which are 500 by 500 microns, and those, again, are used for organoids. We also have arrays that are 100 by 100 micron size rafts and 200 by 200. For all cell types, we recommend characterizing them to see which format you prefer. And then all arrays come in a single reservoir or in a quad reservoir where you can do different medias, drugs, or even different cell lines within the reservoirs.
It's probably worth mentioning, just as supplementary to that, is our R&D team have spent a significant amount of time over the last couple of years characterizing, over 100 different cell types. And we have collated that information, freely available to our customers, so that we can minimize the amount of optimization and characterization you have to do up front. So certain cell types like a little more space, and others don't need so much space. So we have a lot of that information characterized, so we can really hit the ground running with you when you have the instrument.
I just wonder, can you also transfer to 384 plate?
No, not at this time. Only 96 well plates as well as PCR strip tubes and a PCR plate. Yes.
So down the road, is there any possibility to use this for UHTS screening?
For what kind of screening?
High throughput.
High throughput screening.
So we get asked this a lot, right? Can we do isolate into 3D4 well plate? I think a lot of it can be mindset oriented, which is a lot of people think you have a 96 well plate with a clone or an organoid, and that a percentage of them will not survive. But with the way and the timing of when we isolate, basically every well of a 96 well plate is going to grow for you. So there's far less loss of targets. And so you can also isolate multiple 96 well plates from an array. So whilst we know we can't directly deposit into a 384 well plate, you will have hundreds or indeed thousands of clones in a single array. And so the strategy there would be, we can isolate you as many 96 well plates as we have clones on the array. So it's two paths to the same end, but no, not directly into 384.
Thank you.
And the lady behind.
Hi. So for cells in 3D, while they're in the array plate, how do you keep them from floating up and away during culture time when you're exchanging media and bringing it back and forth to the instrument?
Yeah, that's a great question. So the workflow for seeding organoid-forming cells on the array, we're using a methodology that the array is cool, so that we allow the ECM and the cells to settle into those microwells before polymerization. The array then goes into the incubator. That ECM polymerizes and sort of holds those organoids down into the microwell. So it's not as much of a formed layer as you might be used to with domes or layers of matrigel. It's much of a looser matrix, but you can still pretty easily do media changes from the array without disturbing the organoid location. So with these iPSC differentiation protocols, they require full media changes for each of these processes. For your adult stem cell derived organoids, usually I just recommend to our customers to do more frequent half changes, just so they're not, potentially risking the disturbance of the organoid matrix layer.
Thank you. Very interesting talk. I wonder if there's any recommended automation steps that we can incorporate for matrigel dispense for the rafts, because that step can be quite tricky.
Yeah. So currently we're not using any fluidics or any sort of dispensing methodology. The workflow is much simpler than your dome, right? So when you're pipetting a dome, you have to make sure it's right in the center of the well, so you don't have organoids traveling to the edges and things like that. Because we're seeding in an ECM type suspension, the workflow is really straightforward. So the fluidics wouldn't necessarily help us because the workflow is already very streamlined itself.
Also, we're not dispensing into each of the individual microwells one at a time. It's a reservoir into which we dispense that volume, and it distributes itself evenly.
So I guess if there is a recommended volume for different sizes of raft, that's
Yes.
something already figured out.
Yes.
These guys have really well-established protocols that we freely share with customers who are adopting that particular strategy. Yes.
Can you comment on the cost of the machine and the cost of the arrays?
Absolutely. I think that's the only reason they have me up here, is to answer that question. It's very straightforward, actually. And so to have the instrument as you have seen it and to be able to do everything that you've seen Allysa talk about today in the US is $175,000. And the arrays themselves, depending on the application we spoke about, there's quite a number of formats, but they range from $350 to $500 per unit, so per array. The $500 being the one you would use for organoids. But I think it's really important to stress that even with the organoids, we're talking hundreds of organoids per array. And if we go down to the two-dimensional culture for adherent and suspension cells, we anticipate thousands of clones per array. So it should be pennies on the dollar when it comes to per clone or per organoid. Gentleman up here, actually.
Thanks. Yeah, so you mentioned with the organoids, the raft can kind of stick around or go somewhere else in the well. If you're doing suspension cells and growing them to confluence and then passaging later, how do you make sure that you don't keep carrying the raft forward? And then also curious if you have any quantification of ghost wells with using this for cell line development for suspension cells.
Yeah, so about the raft, it's a very small piece of polystyrene. As you're passaging the cells, trypsinizing them, or just moving them as suspension cells, the raft will eventually just get aspirated and be removed. And then, I'm sorry, can you repeat the question about the quantification?
Quantification of ghost wells for cell line development with the suspension cells? Like how often do cells accidentally, disappear, which means they probably moved into another raft on accident somehow?
You're talking about on the array? Or,
Yes, on the array.
Yeah, so, with every suspension cell line, we usually recommend our customers do some plate-based coating optimization. So testing multiple different coatings, sometimes it's coatings in combination. But usually once we figure out the optimal coating material, PDL, PLL, PLO, or some sort of combination, and know how long that coating keeps the cells stuck, we can really optimize the experiment to eliminate any movement of cells around the array. But the other side of it is, because of the time course imaging that you're able to capture with the air system, you have a catalog of clonal conformation, and when you're building those populations in cell raft cytometry, saying, I know the doubling time of my cells, and I want one cell at this time, and then at your second scan, you've hopefully scanned the array at an appropriate time where you expect one doubling and then another. You can really use cytometry to eliminate any concerns of having that single cell, and now all of a sudden it's 4 or 8 cells when it shouldn't be. Does that make sense?
Do we have any other questions? Yes, sir.
So it's the $175K box and the $500 consumable, and that's the pin box system. There isn't additional consumables or?
No. So with respect to the consumables that you have to buy from us, it's purely the arrays. Of course, you're going to have your regular reagents and the ECM and what have you that you would normally be using. It's worth noting that in our arrays, you're using significantly less of those reagents and lower concentrations of expensive ECM. So that's an aside. But with respect to other ongoing costs, it's like any piece of capital equipment in your laboratory that you rely upon. You will most likely take out a service contract on it. We don't do anything weird or different to anyone else. It's an annual service contract that gets you your preventative maintenance and indeed support and coverage for anything should something go wrong. But beyond that, what you see is what you get. One thing I will say is that at present the software, this is what makes it great for multi-user environments in a lab, is that because you can take the software and put it on the computer at your desk, for example, you can take your data and analyze it away from the instrument whilst your colleagues are currently running their arrays and what have you. But we do also, we have an installation and training charge, but the reason for that is it's a full day of field service engineer time to install and calibrate, and then you get 3 full days on site with one of our field application scientists working with you to train you. So, but beyond, there are no other ongoing costs, no. At the lady, and then I'll come to you.
So I was wondering for the organoid, on this example, you start seek So for the organoid, on this example, you seed by single cells, but
Yep.
for the adult organoid, do you also seed
For
by?
the adult stem cell derived organoids, you can do single cells or you can do fragments of cells. If you're not interested in clonal organoid generation, you can take your dome of organoids, mechanically dissociate them into fragments, and put them on the array. And then, of course, you're going to get from fragment to organoid much faster on the array, and ultimately be able to do your downstream assay, whatever it is you're interested in, much quicker than this time course is showing you.
So do you also see huge variation in terms of the size if you control the cell numbers to start with?
Yeah, so when you're doing fragment-derived organoids, you're absolutely right. There is going to be more variation in size. Typically, you can do your dissociation of the organoids in your plate, and under a standard microscope in your plate, kind of look at the size and the uniformity of those fragments, and that will get you a more homogeneous population on the array. But again, the beauty of the array is you're able to assess that population and say, if I'm doing a downstream toxicity assay that's going to be very reliant on total cell number, I showed you 300 to 500 microns as a window, but you could be even more strict and say, I only want organoids that are 350 to 400 microns to even more tightly control your experiment. So the heterogeneity on the array can be ameliorated by your upstream efforts to get your fragments to a normal size. And then, of course, you're using the array as that sort of selection tool for your downstream application.
Ultimately, what it lets you do is create in the 96-well plate a customized screening array of pretty standardized from one well to the next organoid, because you're selectively, as Allysa said, selectively releasing and isolating rafts that meet the parameters that you have set. Absolutely.
Like clonal generation or cell line development, or?
There are, yes, there are multiple strategies, some of which include growing, seeding the cells onto a semi-solid medium, letting them grow to a visible colony, which is then physically disrupted or scraped off to do that. That's one way to do it. It's like any method; it has its limitations and its drawbacks. So ultimately, I think the take-home message for here, with respect to colony isolation, is that this is the least disruptive way to do it, but also because of the statistical nature of our array, it's orders of magnitude higher efficiency, and because we're isolating a colony, not a single cell in a well, the post-isolation outgrowth efficiency, compounds upon that. Yes, it's a great alternative to things like manual limiting dilution. We have an example where in our hands we would isolate something like 2,300 clones from one array, but in order to achieve the same number of clones with manual limiting dilution, you would need 596 well plates and 5 liters of media compared to one array and 5 mils of media. So it's that order of magnitude of efficiency as well. Yes.
That depends on organoid type. For the adult stem cell derived organoids we're usually using some combination of trip LE, sometimes with DNase-1 just to continue to encourage those cells to get into single cell suspension. It can be difficult, and some organoids are inherently stickier than others. But again, to sort of reiterate, part of the beauty of the imaging and the array technology is that you're very easily able to identify rafts that have a single cell versus rafts that have 2 cells that are stuck together, right? And so, the dissociation upstream can take a little bit of tweaking depending on your organoid type, but that's sort of an upstream optimization to moving to the array.
I'm sorry. So how many microwells per array are we?
Well, it depends on format. So the organoid array, which is our largest cell raft size, comes in a single reservoir. It has over 2,100 positions. The smallest format, which is the 100 by 100 micron, has over 40,000 cell rafts in one reservoir. And then depending on the cell raft size and our quad reservoir format, your number of microwells is in that range.
Air systems on the market?
Gary, would you like to answer that question, or how many cell raft air systems do we currently have installed globally?
There's over 30 currently installed in the U.S. and about 10% or so of those are in Europe.
And the composition of that would be 80% of those customers are biotech companies or pharmaceutical companies, and the other 20% would be academics. Again, how long has the system been available? Maybe you could tell a little bit of the story.
Sure, yeah. The company was formed in 2010. Our first commercial sale of the air system was in November of 2018. So we beat the pandemic to get out into the market. And that system was sold to Einstein College of Medicine. And so we started off on the academic side, but then in 2019 we significantly grew our user base, continued to sell through the pandemic, and since we're now post-pandemic, I think we all agree, we've been continuing to push our install base up, as well as increasing the number of applications and cell lines that are supported on the platform. And as Scott said, about 80% of our business is currently pharma biotech.
The lead time on the instrument, it's 2 to 3 weeks at the moment. Obviously, if you all place an order on the same day, it might take a little longer. But yeah, at present, we're looking at 2 to 3 weeks to ship the instrument to you, and then of course it's very schedule dependent. Your schedule, our schedule around the training and installation. But compared to some of the lead times on products within the industry over the last 2 years, we're very quick. Let's put it that way.
Yeah, we don't have supply chain issues. It's available.
And we manufacture both the instrument and the arrays in Research Triangle Park, North Carolina. So from that perspective, everything is here within North America, and we're very confident in our ability to supply very quickly.
Post-analysis of the imaging, so after you've actually defined your desired population, what is the time mechanically to seed a 96 well plate?
You mean the time of isolation itself?
Yeah,
Or-
like when it pokes off, if you want to do a 96 full
Yeah.
plate, how long does it take for the magnet to transfer a full plate?
Sure. So for our adherent or suspension workflows, usually around 45 minutes for a plate. The organoid isolations can take upwards of an hour, and that's just because of that ECM provides a little bit more resistance through the process of moving that array out, or the cell raft out of the array.
Yeah, I would encourage everyone, if you want to see the system, the rafts, touch and feel them, 2430. The poking and the picking, which is a thing that we've created in what is called a hype box, understanding what happens in the system, is also on display in the booth. So please come over, take a look.
Thank you, everybody, for your time. We appreciate it and look forward to seeing you at booth 2430.